September 16th, we have Dr. Faizah Bhatti. She's going to be talking about inflammation and angiogenesis and the retina, a novel view of vascular dysgenesis through the lens of a preemie. Dr. Faizah Bhatti is a neonatal intensivist over at Oklahoma Children's Hospital. She's also the associate professor in pediatrics and associate professor or adjunct associate professor in ophthalmology at the University of Oklahoma Health Sciences. Now, 15 years ago, when I met her, she said, "There's surfactant in the eye and I'm going to study it." And I thought, what? There's surfactant in the eye? Well, now she is an NIH R01 funded physician, scientist who is focusing on the role of inflammation and surfactant proteins in abnormal vessel growth in the retina. She also bakes, she cooks, and she's an excellent knitter. Her favorite color is red. So please welcome Dr. Faizah Bhatti.
Thank you, Clara. That's a lovely introduction, and you know me well. You know me well. And yes, 15 years ago, that was a moment when I thought, you know what? Everybody told me I was nuts, and I got into this in my fellowship, and it was one of those things that I was like, there's got to be something here. It makes sense. And I'm hoping that as I tell the story today, I can convince you all that this story actually does make sense. And thankfully, the NIH did see merit in our work, and we did get funded with an R01. And so I'm going to kind of lead you through the story here. And so I do have disclosures kind of at the end, but for the purposes of the recording, I'm going to state I do not have any disclosures to present. We do have funding from the NIH, the National Eye Institute, as well as from the Knights Templar Eye Foundation. And we will be presenting data generated from that funding today. There are no other relevant industrial disclosures. Hopefully someday soon, not yet. Okay, so what are we going to talk about today? Let's see. All right, outline of today. So preterm birth, as we all know, is on the rise. Why am I fixated on the eye? Why do I study the eye? We're going to talk about that. We're going to talk a little bit about the epidemiology of retinopathy of prematurity and why this is such a big problem. And then we're going to kind of go in and remind ourselves of anatomy that we did way back when in med school, a little bit about the eye and the visual system, but really how the study of angiogenesis and the study of the retina really go hand in hand, and why if developmental vascular biologists such as myself, when we're interested in studying the mechanisms of vascular development as well as disease, why the retina is such a perfect model for us to study this. We'll talk about the pathogenesis of ROP, current therapeutic modalities and controversies, why inflammation plays such a big part, as well as systemic vascular disease and ROP. We'll talk about what we do in the lab, of course. We'll show you some data, and then we'll all bring it back to the bedside again at the end. So this is no surprise to this audience. We know that preterm birth is on the rise. This is the 2024 March of Dimes report card. We got a D+, not great for those of us that like to be academically advanced. No surprise also was that the states in the South had the worst rates of preterm birth. Oklahoma, while we may be 50th in education, we were not completely at the end for preterm birth, but we're up there. And you can see here that the states that are that got an F grade are those such as Arkansas, Louisiana, Alabama, Georgia, South Carolina. Whereas the only A grade state up here is Vermont. So we really have to understand not only why preterm birth is happening, and while we're getting good at saving these babies, we really now have to focus on how do we keep these babies from developing long-term complications, such as retinopathy, which we'll go through. So really, when we look at the susceptibility of the developing fetus and the neonate to alterations in development and structural injury. We have factors which are related antenatally and genetically, but then in the postnatal period, we have many hits that the neonate is going to take in terms of sepsis, which can lead to lung injury, brain injury, malnutrition, of course, oxygen toxicity, mechanical ventilation, no surprise, use of steroids. All of these things, while they're important and they keep our baby going, however, all of these things also contribute to this one by one additive hit that the neonate suffers. And ultimately, by the time we get to 37 or 40 weeks, many of the long-term complications that these neonates are going to face have kind of already gotten set into motion. And so when we look at retinal disease, okay, why is it that retinal disease is such a big consideration for both pediatric ophthalmologists, and there's those of us in the neonatal field that have dedicated research in this. We know the risk factors are lower gestational age, low birth weight, and inflammation. Lower the gestational age, lower the birth weight, the more infection you have, chorioamnitis, more infection, inflammatory hits, the greater the risk. Estimated incidence in the US of any stage of ROP is about 70% in some studies, some centers as low as 40, but really can be as high as 72, 74% there. We know that females, unfortunately, this is one of those complications in which females have greater risk, along with lower birth weight and gestational age. And unfortunately, even with the best of treatment, approximately 17,000 babies are blinded by this every year. They get designated as legally blind. Now, when we look at ROP and we look at the most severe form of ROP, and we line it up with all of the other comorbidities which a neonate might face, of course, our healthcare system, we know, is driven a lot by dollars and cents. And so when you look at the incremental cost of the comorbidity in US thousands, you can see here broken up into three groups. We have our 24-weekers in red, 30-weekers in green, and 36-week gestation in blue. And you can see here that the highest cost incurred is for ROP stage 4. And that should come as no surprise, because as opposed to some of these other comorbidities, which may get better over time, ROP can remain a lifelong comorbidity which these infants have to live with. So the Institute of Medicine estimates the societal economic cost of prematurity to be about 26 billion. We know that for a term baby, it's going to be about 2,830, but for a preterm, it's going to be about 41,000. However, there's this great study by Rothschild in 2016 in American Journal of Ophthalmology. And what he did is that he calculated the cost of ROP, not just based on the initial cost of the treatment, however, also the parental burden from loss to wages, cost to society for loss of wages for the infant. And that added on top of the cost of treatment, both in the NICU as well as after discharge, can come up to be over $1 million. And so if we say 15,000 babies at the low end are going blind every year, you multiply that by 1.2 million, and that results in $19.3 billion every year, which may be the cost to society for the most severe forms of ROP. So this is one of the reasons why I say this to the residents, to the fellows a lot, that we're getting really good at saving these babies. But what we do to keep these long-term comorbidities from happening really starts not just at the time of birth, but oftentimes before birth. And so knowing that this is a disease that has its underpinnings in abnormal blood vessel development, we really do have to understand, how do blood vessels grow both in the fetus and in the neonate, and how does this contribute to this pathology? So if we look at the anatomy of the eye, just very, very briefly, this is something we all know. We have the anterior chamber, and then we have the posterior chamber. The anterior chamber being everything anterior to the lens. So the cornea, light passes through the cornea, in through the pupil, which is through the middle of the iris, pass through the lens. Back here, then, you have the vitreous humor, which by 33 weeks should be now completely cleared. Before that, it will have blood vessels that run through it. And these blood vessels are the blood vessels that begin back here with the retinal arteries, and they cross through the vitreous to supply the structures of the anterior chamber. And they should diminish and regress by the time a baby is at term. But in the cases of preterm birth, the hyaloid vessels may not have completely regressed. And then, of course, at the back of the eye is where the retina resides. The retina is a very complex structure. It's made up of 9 layers and almost 100 different kind of cell types, if you look at the subtypes of cells. But what I found most interesting, and it always fascinates me about the retina, is that the anterior-most part of the retina, which is the front here, that's the side that lies close to the vitreous. And so this is where the light hits. Light is going to hit the retina, but it actually has to travel first all the way to the back here, to the choroid and the photoreceptors. And these photoreceptors are what respond to those light signals. Once they respond to the light signals, they then convert that light signal into an electrical impulse, which then gets transmitted through the neuronal cells, which involve amacrine, bipolar, and ganglion cells, which finally coalesce at the anterior part of the eye. And they then form the nerve fiber layer, which is going to run across the surface of the retina, come back to the very back here, and that is what forms the optic nerve. So it's amazing how light has to travel first all the way down, the signal coming all the way back up, and then that is what's going to take the signal ultimately to the brain. What that tells you is that each and every single one of these cells has to function and coordinate in perfect sync for these processes to happen. And so any injury, any scarring, any gliosis that you have can result in permanent long-term damage to these structures.
I know, you're super.
So when we look at retinal vascular development, we know that in the human, the angiogenic process begins at about 16 weeks from the central retinal artery. At 16 weeks, you have your main blood vessels, which start to arise from here. And you can see that by week 20, you've got about a quarter to a third of the retina covered. 24 weeks, about a little bit more than a third here. 32 weeks, you've got about 2/3 of the retina, and it isn't until 40 weeks that the retina is completely vascularized. And you can see that here depicted if you look at the back, so straight on if you're looking at the fundus. So 16 weeks, the number of blood vessels on the surface of the retina, 8 months, and then not until 9 to 10 months do you have complete vascularization. So why does retinopathy of prematurity happen? What is the pathology? Well, we know that in utero, the retina tissue is highly vascularized. There are a lot of blood vessels in multiple different plexes and layers which have to develop in the retina, not just on the surface, not just deep down, but you have multiple layers of blood vessels which kind of spread out like a lattice covering, a lace covering through multiple structures. So the superficial, middle, and through the bottom. And those blood vessels in utero grow in response to IGF-1. So that's one of the growth factors, one of the main growth factors responsible for blood vessel growth, but also VEGF. VEGF is expressed in response to hypoxia. We know that in utero, we have a hypoxic environment. After the baby is born, preterm, IGF-1 before 27, 28 weeks, actually 29 weeks, is coming from the placenta, and the fetus doesn't make any. So if the cord is cut, IGF-1 is gone. Now babies are exposed to hyperoxia, even if you don't treat them with oxygen. Going to 21% oxygen is still a hyperoxic environment for them. And so a combination of both of these factors, loss of growth factors and VEGF, results in cessation of further development of blood vessels. And so what you get here is this zone in which the blood vessels kind of come and they just stop. So everything beyond that is avascular, everything before that is vascularized. However, continued stresses such as infection and inflammation can lead to actual loss of these blood vessels as well. So not only will these blood vessels stop growing, but you can have a loss. However, the retina is continuing to develop. Even the little bit of blood that's there does continue to supply the demands of the dividing cells in the retina. And ultimately, the retinal cells are going to divide and keep growing to a stage where now you have many more cells than you had before, but not enough blood supply. And so what's going to happen now, you get a secondary hypoxia ensuing, which leads to a very rapid rise in VEGF, an abnormal rise in VEGF. And this now leads to the abnormal blood vessel development, which initially develops on the surface of the retina, but can extend both deeper into the deeper layers, but also grow in towards the center of the eyeball, which can then put traction and lead to retinal detachment. So what are the treatments that we currently have in hand? Well, the gold standard is laser photocoagulation. This is performed, we know, under GA or local anesthesia. It's a lot of burn spots. You need anywhere from 500 in the mildest forms up to the 1,400 burn spots, which are going to burn and ablate every single one of those neovascular tufts. And this is done with an argon green 514 nanometer laser. We know that even though burning these neovascular tufts are going to remove those tufts which have the potential to grow to the center of the eyeball, however, that also leads to a permanent decrease of angiogenic factors. And so therefore, the blood vessels that need to continue growing in this area can get impacted. Typically, these burn spots are going to be about 0.25 burn width apart. And while it is generally safe, there are some complications that can happen, including burns, hyphemia, or blood hemorrhages, retinal hemorrhages. Hyphemia is blood in the anterior chamber, as well as rupture of the choroid. So while this is generally the gold standard, in well-trained hands it's safe. However, it is not completely free from complications. We now know that there are many VEGF targets which are out there, and many more that you read about in the literature that are being studied every day. They generally can be categorized into basically three different categories. Antibodies, of which there are monoclonal and derivatives. Aptamers, which are quenching molecules. Oral small molecules, which are tyrosine kinase inhibitors, fusion proteins, which are traps for VEGF, and then other miscellaneous drugs, which are in the process of being studied, such as siRNA to silence VEGF. Basically, the goal is to reduce the signal of VEGF. So either you're going to inhibit the binding of VEGF to the receptor, or you're going to alter the structure of VEGF, the receptor, or the downstream pathways. So, of course, intravitreal bevacizumab or Avastin is the most well-known of these therapies. It's a monoclonal antibody. And while everybody got very excited after Dr. Mintz-Hittner's paper, which in itself was very revolutionary, the idea that you could do an intravitreal injection of a monoclonal antibody which would target VEGF. However, it is not without problems. We now know for sure that there is systemic leakage of intravitreal bevacizumab, which can occur not just 2 weeks, but in other studies. This is the first one that really showed it, that came out of Dr. Sato in American Journal of Ophthalmology published this in 2012, where up to 2 weeks after intravitreal injection, you had systemic bevacizumab, which was being detected. And not only that, but there was suppression of VEGF. We think about it, it's an antibody to VEGF. So if you have an antibody to VEGF now circulating through the system, you're going to reduce your VEGF levels. What's right behind the retina? The brain. So, of course, the brain and the blood vessels and the circulation of the brain is at greatest risk, but you see this decrease throughout the body of VEGF. What do we know about long-term effects? Well, there are 2 studies. I'm only going to show you 2. The first one is out of the Canadian Neonatal Network, where they had 2 groups. One group of babies treated with bevacizumab, the other with laser, and they did Bailey scores at 18 months. And what they found is that those with bevacizumab had a reduction in their Bailey-III scores for PDI and MDI, as well as a decrease, as well as more severe MDI scores as well. The second study, which came out of the Taiwanese cohort, which is in +1, this was basically a parental choice of treatment. So any baby that needed treatment, parents were given a choice. They had 27 babies with laser, 8 with bevacizumab, and 12 that had both. And what they found is that any baby that had bevacizumab treatment had lower scores, both by itself or if in combination with laser, a statistically significant reduction in MDIs and PDIs as well. So we know that whether you treat ROP, you are going to have complications. Okay, so we know that there's peripheral visual deficits, retinal thinning, you can have hyperopia, myopia, strabismus, amblyopia, long-term neurodevelopmental delays, which we see, as well as an increased risk of adult retinal diseases. However, what we are now understanding far better is that even babies who have regressed ROP or ROP that does not need treatment have increased risk of hyperopia, myopia, strabismus, amblyopia, as well as a significant increase in risk of adult retinal diseases such as AMD, macular degeneration, diabetic retinopathy, and ischemic retinal diseases in greater risk compared to their cohorts, age-matched, risk-matched cohorts who did not have ROP as infants. So we know that this is a problem, and the thought is that even if ROP regresses, you can still have permanent changes in the shape and the structure of the eye cup. So we know that all of these studies, all of these approaches are trying to change the course of a disease once it is already developed. However, I believe very firmly that the only way we're going to be able to make any change in ROP outcomes is if we go back and we understand how do blood vessels develop so that we can rescue that abnormal blood vessel development, which happens initially, and prevent secondary ROP from developing. So a very quick snapshot of what we do in our lab: we elucidate the role of inflammatory and oxidative stress mediators on vascular growth and development, both in the retina and the central nervous system. Our R01 funded work looks at surfactant protein, which I'll talk about, and the mechanisms by which SPA mediates angiogenesis, both in glial cells as well as directly on endothelial cells. And we'll talk about some of this data and what these graphs mean here in a moment. We also are testing preclinical models for rescue of vascular arrest. Using animal models, trying to inject SPA to see if we can rescue blood vessel development. We also are looking at redox modulators. We recently completed R21 grant that I had with a collaborator, looking at one of the redox sensors, thioredoxin, to see if loss of thioredoxin in endothelial cells impacted ROP, and if this led to greater vascular abnormalities. We also are looking at ex vivo modeling. We unfortunately know the news now that any grants that have a primary animal model focus are not going to be looked at very favorably by the NIH, and so we have to look at ex vivo. If we can't study human tissue, how can we create the best possible tissue outside of animals, outside of the human system? And so we're looking at 3D printing using endothelial cells and glial cells, and we're doing this in collaboration with partners at the Biomedical Engineering School at OU. We're also looking at mechanisms to try to prevent lipid peroxidation from occurring, and how these interact with surfactant proteins. We're looking at clinical studies where we look at inflammatory stressors such as infections, as well as hemodynamics of the cardiovascular system, and then also some novel studies looking at the microbiome and how the microbiome impacts retinal vascular disease. And then finally, we've recently started collaborating with investigators in our informatics teams to look at the role of language learning models in AI for data warehouse design. ROP is still a rare disease. It's an orphan disease, so we have to have better ways of collecting data cross-institutionally. And so how do we do this using AI? How do we use AI integration in ROP screening and diagnostic tools, as well as risk factor-based algorithms? So what does surfactant have to do with the retina? Well, we always think of surfactant and surfactant protein kind of unfortunately interchangeably, but we have to remember that it is distinct. 90% of surfactant is lipids, but 10% is the proteins A, B, C, and D. Of these, A and D have immune and inflammatory functions. And it's actually a pretty complicated molecule. If you look at just SPA, it's 630 kilodaltons. The mature protein monomer is about 32 kilodaltons, but then it fuses with two others to form a trimer. And then 6 of these trimers are going to come together to form a tulip. So when did we first start getting interested in the idea of SPA being outside the pulmonary system? Well, this was a review paper written by an alumnus, a neo fellow in my lab, who wrote this review paper where we started looking at the expression of surfactant proteins extrapulmonary. So studies, as I was finishing my fellowship, I was actually working in a surfactant protein pulmonary lab during my fellowship, looking at molecular regulation of SPA, genetic regulation of SPA. But I started getting more interested in the inflammatory profile and the fact that many more papers were starting to come out showing that it is expressed in the genital and reproductive system, in the central nervous system, the GI tract, and there were some studies showing that it was found in the eye, on the cornea, nasolacrimal duct, and in the tears. So when you look at what SPA does, SPA can be upregulated in response to infection and cytokines, but also binds to a multitude of receptors, which further then modulate the neovascular response. And we have a lot of different receptors. We do have some candidate ones in mind for the retina, but there's many different ways in which SPA can modulate inflammation in all of these different systems. So the first thing that we did is that we wanted to answer the question, is SPA expressed in the retina? So this was our paper that we published in 2014. This is Ron Hobbs. He was an ophthalmology resident, an alum who was on our paper with us, and he did a lot of this work. He's now a retina surgeon in Utah. What you see here is immunohistochemistry from the mouse. We have positive control in the lung. So green here is SPA. Blue is DAPI, which marks basically nuclei of cells. We showed that it was expressed in the cornea, as had been seen before, but also in the ciliary body. But for the first time, we showed expression along at P0, so the day of birth of the mouse, in the optic nerve head, surrounding blood vessels. And then as the mouse retina was developing, P0 is at birth, P2 means 2 days after birth, P7 is a week after birth. You can see here that SPA beginning on the surface of the retina starts to then dive deeper. And as the adult mouse retina has completely developed, you see that it is now expressed throughout the different retinal layers. We showed close-up views of it around blood vessels. It does not get expressed, interestingly, in endothelial cells, but it certainly is surrounding blood vessels. And we showed that human glial cells, Mueller glial cells, which essentially think of them as like the macrophages of the eye, that they express SPA. We did this by using we upregulated SPA expression using TLR2 and TLR4 ligands, LPS and PAM3 cis. And what we found is this is in human Mueller cells that we looked at the expression of SPA. This is expression of SPA after, this is first column is control. 1 hour after treatment, 6 hours after, you can see an increase in SPA, 6 hours when you give LPS, and at 12 hours, a significant increase after TLR2 ligand stimulation with PAM3 cis. And by 24 hours, these levels start to diminish. So we then wanted to see, what is the meaning of this? Does it truly hold any importance? What's the phenotype of this? And so what we looked to is a mouse model of oxygen-induced retinopathy. This has been well published since the 1980s. You expose neonatal mice from day 7 to day 12 to 75% hyperoxia. This leads to vessel loss. And then from P12 to P7, so the second week of life onwards, to room air, and by day 17, they now have developed neovascularization. And the reason why this happens is that mice, as opposed to humans, actually get their retinal vessel development all after birth. So at the time that they are born, they have similar vascular development in their retina at about P20, of about a 22 weeker is what it's similar to. And so what we see here, these are what we call flat mounts. So we take the back of the eye, the retina, we stain them with markers for blood vessels, and these you can see are the blood vessels on the surface of the retina. We took two kinds of mice. One was a wild type, and then the second was a genetic knockout. We got these from Dr. Whitsett in Cincinnati. These are global knockouts for SPA. And what we showed is at the day 17, in room air, they both looked identical. However, when you expose them to oxygen and you develop oxygen-induced retinopathy, or OIR, which is the proxy for ROP, SPA knockouts, you can see here the white here highlighted highlights the neovascular disease, and the SPA knockout has a significant decrease in neovascular disease of approximately 50%. We then looked at a rat model as well. It's a little bit different than the mouse model, but similar to the mouse, you at P7 see vascular arrest, and at P19 you can see a neovascular disease. What we are seeing here is at P19, a rat retina in room air, and you can see here the neovascular disease along the border here at P19. And what we found is here, the rat model is nice. The mouse model helps you do genetic studies, but in the rat model, we just used a regular rat. And what we showed is that in that first stage of ROP, where the blood vessel arrest happens at P7, we measured SPA levels from the retina. And what we found is that after exposure to oxygen, there was a significant reduction in SPA expression in these retinas. So then we looked at normal development. We said, okay, we see an oxygen effect, but what about normal blood vessel development? This here is how the mouse retina develops the blood vessels. So you see here again at P3, you see, as I was mentioning, the human correlate. By P5 and by P8, you have your superficial layer, which is completely vascularized. So what we did is that we looked at our wild type, those that had SPA, and knockouts that did not have SPA, reared just in room air with no challenge, and we just looked at the area of the retina that is covered in blood vessels. So at P6, as you can see here, you're only going to have about half the retina, about 2/3 of the retina, which is covered. And you see that here in the wild type, but you see the SPA knockout, we saw a significant reduction in blood vessel growth across the retina. So now we see, okay, not only is it in disease, but in normal development as well. We then went to look at inflammatory as well as cytokine and angiogenic markers at the same time point at P6, again from the retina, wild type and SPA knockout. What we found is that while there really wasn't a difference in angiogenic markers, however, there was a significant increase in our SPA knockouts in IL-1 beta, IL-6, as well as in the angiogenic oncomarker c-MYC. So definitely lack of SPA did have an effect on these cytokines. Interestingly, the macrophage microglia marker wasn't different, but again, you have to remember that this is room air without any challenge at all. So the summary so far, we know that SPA is expressed in murine retina. We know that it is expressed by retinal Mueller cells, both in vivo and in vitro. You can increase SPA both in the mouse as well as in cell culture in response to TLR2 and TLR4 stimulation. We know that retinal SPA is decreased in response to hyperoxia, and that absence of SPA is associated with a decrease in physiological development as well, and that you have a change with absence of SPA in your angiogenic oncogene expression. So that was oxygen and normal development, but what about systemic inflammation? So there was this great paper that came out in 2013. Sophie Tremblay is a collaborator of ours, and Sylvain Chemtab is from Montreal. And what they did is that they showed that if you gave a mouse a systemic LPS challenge, so they took mice at day of life 4, they injected them IP with LPS, and then they looked at the retina at P6, P8, and P10. And what they found is that when they looked at retinal area, 24 hours after injection of the LPS, so after systemic inflammation, there's a significant reduction in blood vessel development. So we took this idea and we said, so what about SPA? Does SPA have the same effect as inflammation, or is it distinct? And so this is Nitsanik Kasa, also a fellow alum, now faculty at OU. And when she was working in my lab, we developed the hypothesis that SPA is going to be increased in mouse retina in response to systemic inflammation, and that this was going to impact vascular morphology. And so what she did is that using the same model, we took, however, this time, not just wild type mice, but also SPA knockouts. At P4, we gave them an injection of LPS, and then we looked at the retina at P6, P8, and P10, looking at the morphology of the blood vessels, gene signals, as well as protein. First thing we did is that we showed that 48 hours after injection, so 48 hours after systemic inflammation, SPA levels are significantly increased, more than doubled in the mouse retina in just the wild type. So this is not without deficiency of SPA, just in a wild type mouse. If you look at what happens to SPA, SPA levels are increased. Still a little bit high, 96 hours, but not statistically significant. So then we said, what do you do? What happens if you do a double hit? So now if you have SPA deficiency and you add SI. So what we did is that we looked at vascular development at P4. And what you see here is in light blue, you have wild type animals, so SPA positive with normal saline. Dark blue is SPA sufficient mice with LPS. Pink is SPA knockouts with normal saline, and then red, dark red is SPA with LPS. So this is at the time of injection. So obviously we don't have any systemic inflammation yet. And you can see that the knockout as well as the wild type, both at P4 at least, have similar blood vessels. However, if you now look 48 hours later, LPS as well as SPA challenge both reduce blood vessel development. So this here in dark blue is if you just give LPS, so blood vessels are decreased, similar to what the Tremblay paper showed. If you had just deficiency of SPA, lack of blood vessels was similar, parallel to that if you had deficient systemic inflammation. However, interestingly, now if you have a double knockout, a double hit, the effect is compounded. So if you think that wild type normal saline, which is the normal condition, is 100%, if you give a double hit, you now have a 22% reduction in blood vessel growth. Just LPS reduces by 9%, just SPA deficiency by 12%. We looked at other markers of the number of blood vessels that are there, the density, and what we found is that systemic inflammation leads to less blood vessels, number of blood vessels, decrease in vascular density, and decrease in branch points. And this was similarly mirrored by SPA deficiency as well. And then we looked at inflammatory mediators, 48 hours, and then at P10, and what we found is a significant difference of inflammatory mediators with deficiency of SPA. You have a reduction in TLR4, you have a reduction in TNF alpha, reduction of IL-1 beta, VEGF, and IBA1. We wanted to look at this a little bit more closely, and so we looked at microglia, which are another glial cell type, to count the numbers both in the central area of the retina as well as peripherally. And what we found is that after 48 hours, the total number of microglia that we counted in the retina were decreased, both in the central zone and the peripheral zone. Peripherally, they come back to normal at P10. Interestingly, in the central zone, you actually see more microglia in the SPA knockout. And 96 hours later. However, here's where things got really interesting. When you look at microglia, this is a microglial cell here, and you look at the morphology, they've got a lot of these processes, or what we call the ramified structure. When they're under stress, they develop this rounded amoeboid structure, which is an appropriate response to systemic inflammation. So in wild type with no SI, you see this ramified pattern. When you give it LPS, you see amoeboid. But with SPA challenge, you no longer see this reactive response, meaning that lack of SPA was not allowing the microglia to respond in an appropriate manner to the inflammatory challenge. So we showed that we see SPA is increased 48 hours after injection. There's a difference in levels of inflammatory cytokines. Secondary or chronic inflammation changes a little bit the way that the cytokines go, but you still see a differential expression. So now in our lab, we're looking at what are the mechanisms. So we went back to our P6 room air, and what we did is that we did two tests. We took whole retina and we did RNA-seq, so that is whole genome sequencing, as well as whole proteomic sequencing. These are two scientists. Previously, we had Johannes and now currently Henry. They then took this RNA-seq data and proteomic data, and we combined them together to do what's called ingenuity pathway analysis. And these are our results. We had two different families, which really are of interest to us right now. The first is a family of molecules which are known as chaperones. So chaperones are important for functioning of proteins. They are important for how proteins get expressed, for how they respond to stressors such as hyperoxia, to inflammation, and there's a variety of families. When chaperones don't work well, they get labeled for degradation by markers such as PELI, WSB2, and KLH33. So the differential expression we saw in genes and proteins were related to chaperones, ubiquitination, transport, and then remodeling. So we didn't really know what a lot of these genes did, so we went back to the literature. We were wondering why this family of crystallins showed such a significant change in their gene expression, especially crystallin AB. Well, going and diving a little bit deeper, what we found is a paper in 2010 which showed that you need this crystallin for regulation of angiogenesis by proper folding of VEGF. And this indeed correlated then with confirmatory studies that showed that you have RNA which is getting expressed of VEGF when you knock out SPA. However, the protein is decreased. So what do we think is happening? The Mueller cells express SPA, they secrete it, and then this SPA is important for expression of crystallin. Crystallin is a chaperone, which without it, the protein is not going to get folded. And so what's going to happen is if VEGF remains unfolded, it gets degraded, it gets marked for degradation by the proteasomes. However, if it folds, that's when you get proper angiogenesis. The second family that we found of markers was related to the cytoskeleton. I'm not going to go into the details of these, however, just to say that ARPC2 and C4 are important individual polymers of actin. They need to come together and polymerize for filopodia to form. And so the question that we ask now, and this is a part of our R01 work that we're doing, is that does SPA directly impact endothelial cell actin? And so what we did is that we first looked at cell culture of endothelial cells, and we looked at cells treated with SPA. They seemed to migrate more. They also showed that they proliferated greater. So this here is control, cells treated with control medium and cells treated with SPA. So greater migration and proliferation. And then when we did immunohistochemistry, looking at actin markers, we did this in collaboration with Dr. Margadant, a collaborator from the Netherlands. He taught us how to do this in live cells. What you see here in the first panel is cells that are have no SPA treatment, followed by cells which have increased SPA in a dose-dependent fashion. And what you see here is this white is the actin. And so the more SPA you treat with, the longer the actin filaments, the more polarized they are, the greater they tether to the cell membrane, and the more number of cells you basically have across. So what we think is happening is, in response to these genes, it leads to F-actin branching. The branching then leads to formation of filopodia. So in two mechanisms, both by glial cells, directly as well as indirectly on endothelial cells, we believe that SPA is acting. But at the end of the day, what does this mean for humans? This is a paper we actually just got word this week, got accepted for publication now in pediatric research. The two first co-authors are my brilliant medical student who's now started her OB-GYN residency St. Louis, Kelsey, as well as Dustin, who is a PhD candidate, grad student in Indiana in the lab of my collaborator. What we did is that we know that a lot of studies have shown you can't take human retinal tissue. It just can't be done. We can't study it. However, we know that if you look at SPA variants, we know that they have been shown to impact neonatal lung disease. We wanted to test the same hypothesis in humans to see if these variants impacted retinal disease. So we had a cohort of 59 infants, less than 32 weeks, less than 1,500 grams, who got screened for ROP. We took DNA. We then did genotyping to look at single nucleotide polymorphisms and allelic variants of surfactant proteins. And then we did logistic regression with genetic and clinical factors, including bronchopulmonary dysplasia, gestational age, and oxygen. And what we found is that when you look at these different single nucleotide polymorphisms, there's 4 here in SPA two. We know in humans you have two SPA genes, SPA2 and SPA1. We found 4 variants in SPA2 and 4 in SPA1. However, out of these, only one, the one variant, which is present in the carbohydrate recognition domain of SPA, we found that gestational age was protective when you had the wild type CC, but less protective when you had the non-wild type, so a thiamine instead of a cytosine. Interestingly, though, even though you had a amino acid switch, it did not result in a nucleotide switch. It did not result in a amino acid switch. So we're not exactly sure why that is the case. We also saw a trend in association with an SPD. An SPD variant, 168, also in the carbohydrate recognition domain. We know that more days on oxygen increase the risk of ROP, but only when you had the wild type. When you had the amino acid adenosine, which resulted in alanine in the amino acid, this now lost its protective effect. So what does this mean? This means that there may be some direct effects of these amino acid substitutions, of these changes. When we look at where these are located, we know that when you have differences in the N-terminus, this is a cysteine-rich region. It's important for disulfide bridging and oligomerization. So SPA monomers coming together. It also may be impacting cytoskeleton. Changes or SNPs in the body may be disrupting the folding of SPA2. And then when you have changes in the carbohydrate recognition domain, this changes how this binds to receptors. So now our next goal is going to be really looking at how we can model these changes. I'm not going to go into details here, except to say we've also done other studies looking at thioredoxin and long-chain fatty acids. However, I do want to show one study, because I'm really kind of proud of this. Here. And this is looking at the GI microbiome. So we know that in adults, in an ocular disease, there's this growing body of literature that shows that there's a gut-eye axis, meaning that in adults, if you have dysbiosis, gut dysbiosis, this can lead to an increased risk of age-related macular degeneration and uveitis. So we wanted to test the same theory in neonates. So what I did is I partnered with Barb Warner at WashU, and we did a secondary analysis of her microbiome studies. We looked at 82 preterm infants that were reported in the Gibson and Warner paper of 2016. And what we did is that we looked at the enrichment of just the first stool sample. We now are looking at the further ones, but our first analysis was just looking at the first. And our first study was looking at two things: richness and Shannon diversity. So what does that mean? That means how diverse is the microbiome? The more diverse, the healthier the gut. And what we did is that we then looked at this in association with antibiotic usage prior to collection of that stool. This is our baseline clinical demographics, which, except for, you can see as expected, babies with ROP were lower in birth weight and, of course, had worse disease. What we found is that alpha richness and alpha Shannon, both measures of diversity, trended towards lower diversity, both in babies when you looked at them with and without ROP, as well as a trend when you looked at them with staging of ROP. And we did Wilcoxon rank sum testing, and we found that the alpha Shannon had significant difference in diversity in non-ROP versus stage 2, and as well as in stage 1 versus stage 2. And then when we looked at courses of antibiotics, this had no impact on diversity. So it didn't matter whether these babies were treated with 1, 2, 3, 4, up to 14 courses of antibiotics. As I mentioned, this is the first stool that the baby passed. Unfortunate that some of these had 14 exposures to antibiotics before they passed their first stool. However, didn't matter how many antibiotics they got, there wasn't any difference in this diversity. We're now looking at what is the signature of these. We're also looking at whether there's differences with gestational age, birth weight, and delivery mode. And we did present this data at PAS. I am also leading some clinical studies with the CHNC. I'm only going to show you this graph here. Our concern is that we may be missing babies in our tier 2 criteria. So our tier 2, what is tier 2? Those babies who are greater than 30 weeks or greater than 1,500 grams. We know that the AAP says that those babies should get screened in these situations. However, all ophthalmologists believe that there's not very high numbers of ROP in this group. Working with my co-group leader, Jeff Schenberger and Rakesh Rao, we looked at CHNC data compare tool. We're now analyzing the actual data, but just looking at the compare tool, what we found is, I'm going to show you the graph, that at least 1 in 8 infants greater than 30 weeks who were screened had ROP, and at least 1 in 10 infants who were greater than 30 weeks gestation met the criteria for needing treatment. We furthermore found some sexual dimorphism, seeing a greater ratio of females in those that were of the higher birth weight group. So again, a lot of things at play here. Systemic inflammation, surfactant proteins play a role. What are these roles? Do they work together? Is there one master switch? Highly unlikely. We are now looking at things like artificial intelligence to see if it can help us both with diagnostics in terms of language learning models for diagnosis of ROP, but also how it can help us in diagnosis using AI enabled retinal screening tools, both fundus images as well as things like optical coherence tomography, which is essentially 3D ultrasound of the retina. We recently got 2 grants working with our data informatics group to help us build a data warehouse. And what we're doing is that we just tested artificial intelligence head-to-head with my medical student with extracting data from the charts. No surprise. The language learning model did it faster. The accuracy is actually pretty impressive. We're still tweaking it. We're still working on it to see if we can improve the ability of it to extract data. But really, the goal is twofold. We need to be able to write programs which we can use to extract all sorts of clinical data and use then that program across different institutions, and then take that data which we extract from different EMRs. So if somebody's using Epic, if somebody's using Crib Notes, if they are using Cerner, have that data get standardized by the LLM and get fed directly into a data warehouse. And so that's really the goal of the 2 grants that we got right now, is to see if we can develop models like that. Of course, this work takes a lot of people, a lot of dedicated people in my lab over the last 14 years. Currently, Henry Song, our staff scientist. We have a postdoc, David Mondaka Ruff, a neonatology fellow, SIMA, and another staff scientist, Sabiha. Our collaborators, Leiden University cohort's been so helpful in helping teach us the ex vivo modeling with our 3D bioprinting and trying to develop extravascular channels. Collaborators at Penn State, which is my alma mater. It's where I did my pediatric residency and fellowship. It was actually in Yana Flores's lab that I started working on surfactant protein. And Zisas Kronios, who's still at Penn State, is a co-investigator on my RO1 with me. Of course, our collaborators, past and present at the Center for Pregnancy and Newborn Health. We are now known as the Neonatology Research Group at OU, University of Oklahoma Children's Hospital, Pete Vitiello, my co-investigator on my R21, Lynette Rogers and Trent Tipple. Trent, of course, our fearless leader. Barb Warner at WashU, the Vision Research Group at University of Oklahoma. Invaluable colleagues, Oklahoma Medical Research Foundation, the vascular biology group there, led by Courtney Griffin and one of her scientists, Chris Schaefer. My first mentor in ophthalmology here at OU, who is now at University of Florida, John Ash, taught me how to do retina dissection from a mouse, taught me how to do staining, and helped me get those first beautiful immunohistochemistry pictures. And then Patricia Silvera, Cannot say enough thanks to her. She and I were both together at Penn State. She was a postdoc, PhD from Argentina. When I was a fellow, we both trained in Yana Flores's lab. She is now at the University of Indiana School of Medicine, and she was my collaborator on the paper that we are just publishing on our surfactant protein variants. And it's her grad student who co-first authored the paper with Kelsey with me. And then, of course, Lois Smith, one of my dearest mentors from Boston Children's Harvard. She is actually the lady who first developed the mouse ROP model. So kudos to her. She has done years and years and years of work on developing and standardizing that animal model. And then, of course, as I mentioned, our funding sources, without whom none of this work would have been possible. We're currently funded through our National Eye Institute R01. We have a Knights Templar Eye Foundation clinical grant for our AI project, as well as a University of Oklahoma seed grant from the AI initiative. Takes a village. So many people I've not been able to mention, friends, both current as well as in the past, my family up here in the left, my father who passed away last year, our children and my two nephews, my husband, partner, Chuck, current lab members here, and then folks through the years, people who have been friends and, as I said, mentors, colleagues, the best anybody could have ever hoped for. We have to work together, everybody. That's going to be the last pitch I put in. We have to do multi-center studies better. We've got to get together. We've got to learn how do we combine our data? How do we standardize our data? How do we learn from each other? There's more to be gained when we collaborate and share data and research than when we try to work in silos. So with that, I thank you. Thank you to Dr. Song here and my absolute dearest Liana here, 15 years, still going strong. So thank you for the invitation.
Thank you so much.
It's been an honor.
Thank you so much, Faizah. That was a ton of information to go through. Yeah, you're already getting claps. Please put your questions in the chat or the QA here, and we'll have at it. But I'm gonna start. We already have one question in there. So I have so many questions actually, but one of the slides had said the DHA supplementation is not enough, right? That early studies from Lois Smith had found that association, and perhaps giving mothers more DHA or babies more DHA, or through supplementation of lactating moms, that could possibly be a prevention model for ROP. What are your thoughts there?
So that happens to just be one of the slides that I kind of raced through, but because the question came up, I'm going to go back and I'm going to show you. So this study was actually a very interesting one. We got 2 foundation grants to do this work, and the whole premise is that DHA, like you said, is so important for neuronal tissue and for the retina. So when we look at DHA, it's a long chain polyunsaturated fatty acids, and it makes up about 50% of the plasma membrane of the neuron. It's involved in, it's a specialized it's involved in release of specializing pro-resolving mediators, which are important for inflammation. And the retina, we know, is highly enriched in this PUFA. So the whole thought is that if you expose the oxygen, the retina to oxygen, you're going to have lipid peroxidation. And this can then lead to breakdown of this DHA. So there's a company that we are working with, and what they did is that they've developed a proprietary molecule, which is a DHA that resists lipid peroxidation. And the way that they formed this is that HDHA here is the normal DHA, and so what you have here is hydrogen here on your methylene carbons. And what they've done is that indeed.
Are you showing us a slide? Because we only see you.
Oh my goodness. Oh my gosh. Okay.
I feel like it would be easier. We can try to follow you.
Share this again. Where can I share this again? Window. Here we go. Perfect. Okay, there we go. There we go. This happened before. So the way that this DDHA works is this is DHA, and you see the hydrogen molecule. So what it does is it substitutes each one of these hydrogens for a deuterated a deuterated molecule. So a deuterated molecule here, and what that does is that it stabilizes the DHA. It keeps it from breaking down. And so what we wanted to do is that in our first models, we wanted to see that if we fed dams, so pregnant mice, if we fed them this deuterated molecule, could we get it to come through breast milk? Could we get it to get into the systemic circulation? Can we get it to go into the retina? So this was a fellow who worked in my lab, Victoria, and so this is our model. So we fed the dams with either normal DHA or the deuterated DHA. Oh, going back here. And then what we did is that we looked at their retinas at P12, P17, again, the vasoobliteration and the P17. We looked at enrichment of DHA, and then we looked at disease here. And some interesting findings. First of all, yes, we were able to detect DDHA in the breast milk. And this is at P7. Just looking at the retina in room air, we found that almost greater than 90% of the retina had enriched deuterium in it. So we were able to replace the regular DHA with deuterated DHA. So we were very, very excited. The next thing that we did is that we looked at our disease. So white is a diet that completely lacks DHA, yellow is normal DHA, red is deuterated DHA. What we found is, much to our dismay, that while avascular area, seemed to be reduced with the deuterated DHA. So the first stage, you had greater blood vessels. So the opposite of avascular, greater vascular. So in the first stage at P12, however, at P19, at P17, not only was there no effect, you actually might have had worse neovascularization here. And so this got us stumped, and these are the retina flat mounts showing you what that deuterated DHA, you saw that there was far greater number of disease. So we were like, what the heck is going on? So we looked at markers of lipid peroxidation, such as T-bars. T-bars are a molecule that you can measure in both the blood as well as in the retina to see, are you getting oxidative stress? And in the red here, again, is the deuterated. So while each one of these had less T-bars, still did not have a protective effect. We then went and we looked at levels of deuterated DHA, and in the retina as well as in the blood, we actually had much higher levels of total DHA. However, it then hit us, and we're now in the process of evaluating the data further, that while you're able to rescue DHA and while you may be able to keep DHA from breaking down, if you do not do the same for AHA, arachidonic acid, that is what causes the imbalance. And so if you only supplement DHA without supplementing AHA, you actually have greater inflammation. And so now what we're doing is the company, they've made a double molecule where they have stabilized both the AHA as well as the DHA, and that's going to be the next step in seeing if we give that to the dams, if we're able to see any protective effect.
Oh,
so I hope that answered your question.
Yes, 100%. Thank you.
Well, at least we know that if you feed it to the mom, it goes through the breast milk. So the whole goal was maybe we can give this as a supplement during pregnancy, right? You don't know who's going to deliver prenatally, but if it's safe in the mom, which we showed it was, perhaps this could become a part of supplementation, but we obviously still have a lot of work that we need to do on this. But very, very interesting data nonetheless.
We have a question about the range of gestational age that SPA is critical for retinal development and or involvement.
Yeah, so very good question. And actually, this is something that a lot of our reviewers asked us when we look at our data. And I think that I can show that best in this graph here, going back. So what we did is that we looked at not only the effect of LPS on retinal vascular development, but we also looked at the effect of SPA deficiency. And so what we did is that room air reared mice, going back here, I wish there was a way that I could stop. Here we go. So what we did is that we took mice from P4 to P10. So essentially by P10, the retina here is completely vascularized. So for a moment here, I'd like you to ignore the dark blue and the dark red. So imagine the dark blue and the dark red are not here. What you see here is that at P6, just by knocking out SPA, which is the pink here, you had less blood vessel development. However, by P8, blood vessel development has completely caught up. Okay. We think that that is because while SPA is playing a role in blood vessel development, it's not the only factor. There's also things like efferents, there's VEGF, there's hypoxia-inducible factors, there are microglia and macrophages which are getting expressed. And likely in this global knockout, other factors came to rescue further out. However, we have now developed a inducible model. We Actually, I might have a disclosure slide for next time because we're applying for a patent. We just developed a floxed SPA mouse. And so what does that mean? That will allow us to not just do a global knockout, but a targeted knockout in cell types of our choice. So we have mice that you can put a marker for an endothelial cell, and using the flox mouse, you can knock out SPA in just the endothelial cell, or in just the Mueller cell, or in just the glial cell. And so you can also do that at different ages. So you can do that at P0, you can do that at P4, you can do that at P10. So these are called inducible knockout models. And so now that we have this knockout, we'll be able to look a little bit closer using the Mueller cell Cree to knock it out of the Mueller cell at these different time points and look at a specific effect, not a global effect of lack of SPA, but a specific effect in just that. But no, that's a great question.
So to help us understand, in the mouse model, P4 versus P10, where would you kind of parallel that in gestational age for our preterm babies?
So going back here to the slide I showed you before. So here we go. So P3, as I said, birth is at about 22 weeks. P3 is equivalent. So you see here with the amount of blood vessels here, the red are the blood vessels, the green are glial cells, astrocytes. Astrocytes develop before blood vessels do. So in the mouse at P3, you've got about a third of the retina covered. By P5 at about half. But this is just the superficial plexus, and by P8, you have the complete superficial plexus vascularized. So P8 here is equivalent to about 36, 35 weeks. Actually, probably about more 34 weeks because you start to get deeper blood vessels. But complete development, which occurs between P14 to P21, is equivalent to about postnatal age 1 to 2 months in the human.
Okay. That's very helpful. So 22-ish weeks at P0, and then at P8, we're at 34 weeks. We're late preterm.
Yeah, exactly.
Sort of. Okay, now we're going to end on a clinical question here. So what are your thoughts, Dr. Bhatti, on the saturation limits for our preemie infants? Like some have suggested lower saturation limits. Yeah, hot. Or for a certain gestational age, and then perhaps increasing the limits to help with ROP. Not a hot button topic whatsoever.
I'm going to start with the general that we do not know. Okay, we do not know. I think that we have to be very careful with the amount of oxygen that we used. The less oxygen we can use initially, the better. I know that there was a lot of interest around the idea of raising oxygen around the time of 32 weeks, so later, as BPD is starting to develop, to see if creating hyperoxia can lead to decrease in VEGF, because we know that when you have hypoxia, you have greater VEGF expression. So if you have hyperoxia, you're going to have less VEGF. And so is there a time period when you can change how much oxygen you're using, exactly as the question is? And I don't think any of the clinical studies have really shown any big benefit there. I believe that oxygen by itself is not going to be the answer, because all of these babies in which we are trying to modulate oxygen levels are having changes in their hemodynamics as well as their systemic inflammatory parameters. And so that is why I'm a big believer in not trying to look at the organ in isolation, but really looking at what's happening to the rest of the infant. I think that by the time ROP develops, the damage is done. There's nothing you can do. So the question is that how early can we try to rescue blood vessel development? And really, I mean, we don't know what that right number is yet. So I don't think oxygen is going to be the answer. I think it's going to be by modulation of other vascular growth factors, trying to rescue blood vessel development.
Yeah, to dovetail on that, what do you think is the most hopeful modality or avenue thus far from your research that would help us with this prevention?
Honestly, I think it's being very mindful from the get-go about nutrition. I think adequate
Mhm.
nutrition has probably been by far the greatest, has had the greatest impact on reducing ROP.
Mhm.
I think when we look overall at all of the different things. Now, the question is that so many of these inflammatory mediators, my other pet peeve, I'm just going to say this right now, is that we tend to label mediators, inflammatory mediators, as pro-inflammatory or anti-inflammatory, and people think that way about SPA as well. It's not a question of is it good or is it bad, but it's how much do you need at a particular time point. And so I think it's going to be, how do we safely upregulate these growth factors and these immune factors, which are critical for development early. And so I think it's with SPA, can we deliver it systemically? Can we find ways to deliver it to upregulate its expression beyond just, I mean, surfactant formulations don't have SPA, but it's not just in the lung. We're going to have to find ways of doing this, because I didn't show the data today, but there's a lot of interest around brain. And the identification of SPA around the choroid plexus and the germinal matrix. We have data to show it's there. Others have shown it's there, and that there's upregulation of SPA with inflammatory diseases such as encephalitis and meningitis. So can we find key mediators which can be upregulated early on to be able to maintain normal development?
Yeah, the key question. We have one late breaker question, and we'll end on this. So based on retinal development, is it could it be something that we want to think about adjusting ROP screening, the time at the gestational age that we do that?
Great question. I believe that, I mean, everybody wants to screen less. I get it. That's really the goal. How do you modify screening criteria? How do we screen less babies? How do we get better at detecting the ones that need to be done? I think that there's a lot of good evidence to show that there are many now growth-based screening algorithms. In fact, we did a retrospective analysis, and we published this 2 years ago, of the GROP criteria, the growth and retinopathy of prematurity criteria. So how do we look at postnatal weight gain and use that to determine which babies should get screened or not? However, that should be looked at completely independent of the fact of post 30 weeks, post 32 weeks, post 1,500 grams. Are we doing a disservice perhaps by not screening all of the babies that require screening, especially knowing that 1 in 10 babies are going to require treatment? And I think that we need a different approach. And that's one of the things that with the CHNC focus group and with the clinical collaboratives that we're forming to study this disease that we're trying to look at, is it's not going to be a same screening protocol, I think, that we can use for the two.
Thank you so much. That was a ton of information. We really appreciate it. There are a lot of maybes, and that's why it's so important for babies to have science. So thank you for doing the hard work and then giving that information to us so we can work at the bedside. Dr. Bhatti has allowed for us to record this session. We will have it available for guests and internal use. Remember to do your survey and come and join us next month. We have Dr. James Wynn talking about neonatal sepsis. Thank you so much, Faizah.
Thank
Have
you.
a great day, everyone.
Good to see you guys. Thank you all again.